Our next speaker today, I wish I could say this with a French accent, but I can't. Emmanuel Mongodin from University of Maryland School of Medicine. Here you go.
Thank you. You have a pointer? Oh, that's okay. I can do it. Well, good afternoon, everyone. Well, I would like to thank the organizers for having me today. And so this was the title of my talk. And when Lynn and I discussed a few months ago, this, was a good idea for me. And I thought that, it would be something great to talk about. And then when I saw that the session was on microbial exposure, so I decided to okay. Is it oh, there it is. I decided to basically add a subtitle and maybe switch the focus a little bit, and hopefully you'll understand why in a little bit. So what I wanted to talk about and maybe emphasize is the fact that we basically produce a lot of data. And as Jonathan mentioned earlier today, this is by no means a field that is brand new. So people have looked at microbes in general and the built environment in particular for a long time. But ever since the rise of metagenomic strategies, we've seen the field, expanding dramatically, and as a matter of fact, it correlates nicely, at least in the number of publications, in the steady increase of publication basically related to microbiome. So the question now is, so that comes from, I think, a really nice review that was published last year after this meeting, I believe. And the question now is not so much what have we learned about microbiomes of the indoor environments, but basically how do we apply that research to change how we design, build, and operate buildings. So basically how we translate that into either different strategies to build or to clean, or basically make us live healthier. So obviously I'm preaching to the choir here, and this point has been shown over and over again. The microbiome impacts pretty much everything and anything, and not just the human body, but also the environment. The point I'm trying to make here is that we have all these processes, and again, here specifically for health, but it applies to pretty much everything. But most of them are basically associations, and there are only a few things that for which mechanisms have been identified. And so the question now is, how do we translate that into causality? So for human microbiome research, how do we change the microbiome, whether it's in the environment or whether, so basically what we're exposed to, how do we translate that into basically ways to make us live healthier? So what we all do is those hypothesis generating experiments where we basically look at associations between various types of environments, so whether it's human-based or whether it's things that we swap from the environment, and look at associations with different molecules, microbes, and other biomarkers. Now, part of the exercise is to then do some evaluation and validation of these hypotheses using in vitro or in vivo experiments, and basically pinpoint to whatever culprit we think is responsible for a specific condition. When we do a survey like this one, I keep doing that. So this is something that, some sampling that Lynn and I did not too long ago. So that was in the spring as part of Chris Mason Metasub subway system, Metasub project. And so that was actually in Baltimore, and a piece of advice here: if you want to have a good press coverage and generally positive, make sure you have the mayor coming with you. It's always quite positive. But part of this is also to educate people about why it is necessary to do this survey and what we're hoping to learn. The key here is we have to learn not to be such germophobes and realize that some bacteria is good for us. And I like that quote here: I take tourist transplantation all the time, so despite the fact that subways are dirty, and I never get sick. So I think that, and that also emphasizes what Jen said earlier, that obviously when we take the time to explain to people and also make sure that journalists quote us appropriately, then I think that there's something powerful that we can convey to people here. The problem is when we start presenting data, and that's when things become a little scarier. So again, these are some slides from Chris Mason, and so these represent just some of the preliminary data from the various cities that were sampled over the past Metasub sampling days. So Washington, D.C. and Baltimore are among them, but there were many cities across the world. And this is just a heat map of antibiotic resistance genes that were detected in various subway systems, and here just a list. And the problem here is that when we try to explain or go back to either local authorities or even people that are asking us on the subway what we find, then this becomes very, very scary. And then, as Ed Young pointed out yesterday, we end up in these situations where we have those headlines where having a cell phone that has more bacteria than a public restroom is kind of gross. So we have to tell the public, well, yes, it might be gross, but it's actually probably okay. So the mixed message after that comes when we are trying to basically tell people that, well, it might be gross, but there are some things that are actually required for health. So this type of behavior or this one, so that was actually my daughter when she was probably 4 here, and so far she doesn't have any allergies, and I think that, you know, we thank that poor cat for taking all the beating to keep her healthy. So this is actually good, and that what we find logical about trying to clean as much as we can is probably not a good thing. There was another book that was published earlier than Ed Young's book that nicely summarized that. So Marty Blazer's book, talking about how sanitizing our environment is probably not a good thing, and that the rise of antibiotics have created issues that we never anticipated before. The problem here is that, as usual, when headlines appear like that in the media, then things become a bit mixed messages. So it turns out that not all the germs are bad, and even some bad germs are not all bad. So the question is, how do you take that information and use it to, for example, change policy? So the reason, so this is where I wanted to present that project. So that doesn't have much to do with the indoor microbiome, although just a little bit, but this is kind of a cool project for 2 reasons. So what we do here is basically looking at the bacterial communities associated with tobacco products. So the goal here is not to make cigarettes safer to smoke, okay? We know that cigarettes are bad and that will probably never change. The goal here is to develop new ways to regulate tobacco products. And the reason for that is because the tobacco industry seems to always be 10 steps ahead of regulators. So something that people don't know is that cigarettes are like cheese. They're living products, and basically there are bacteria everywhere. So there are bacteria in the tobacco itself, and depending on the type of tobacco that people use, there are different sets of bacteria. Some of them are going to be plant-associated, so I'm not sure if you can see, but this is Pseudomonas, and others that we think are there because of the manufacturing process, and that's the case, for example, for Staphylococcus. The problem here is that first, so these bacteria, so we find them on the tobacco, they don't get burned, at least not all of them, when the tobacco is smoked, and we can find them, so I think this is a bit misleading, not on the second-hand smoke, but on the mainstream smoke, so the smoke coming here out of a cigarette, and we can collect those smoke particles and put them on Petri dishes and basically grow these bacteria. What you're going to ask me here is, well, why does that matter? And that's exactly what the tobacco industry told us. Bacteria are everywhere. We are always exposed to bacteria. So why having bacteria in the tobacco, you know, what does it matter? Why do we need regulation for that? What people don't know is that some of the carcinogens in the tobacco products that we call TSNS for tobacco-specific nitrosamines are the results of bacterial activities. So that means that the more bacteria that you have, the more carcinogens that you have. And these processes are completely unregulated, and as a matter of fact, they're not only ignored, but they're also completely unknown. So we don't know which bacteria are involved in creating those carcinogens, and therefore trying to regulate that. Because when you buy a tobacco product, when you buy, for example, a cheese or some meat, the bacterial load in these products are checked. It's not the case for tobacco products. The problem here is that we get a lot of push, and we basically have to say, well, how do we know that these bacteria are bad for health? So we go back to the diversity that Marty Blazer talked about, and that sometimes diversity is a good thing. And here, so this is just a network analysis showing some non-users, and here people using different tobacco products. And as you can see, so there's a lot more core bacteria in tobacco users than non-users. So in this case, basically bacterial diversity is not a good thing because we know that these bacteria are going to have deleterious health effects. So what we are trying to do here is to use this information to basically give ammunition, if you want, to people that are doing regulation and use that information to find new ways to regulate these products. This is also a powerful message for smokers, and I guess it goes against what we're trying to do when we're trying to say that not all bacteria are bad. If we tell smokers that their products have a lot of chemicals in them, they basically don't care. If we tell them that there's a lot of bacteria and actually way more than a toilet seat, that's a really strong deterrent for them to stop smoking. So maybe that's something that in some cases can be useful, that growth factor to basically reinforce that message. So where do we go from there? So obviously, so what I was trying to say here is that the situation is complicated, and using that information is not always straightforward. So what I wanted to finish with is highlight a project So I'm not going to present results because my collaborator, so Amy Sapkota, so she's also my collaborator on that tobacco project. And so she's the brain of that project. So, so she would be definitely better than I would ever be to present the data for that project. But what I wanted to do was to highlight that project as kind of like maybe a framework to how we can take that information and use that information to basically change policy and how we deal with various problems. So in this case, so this project is called Conserve, and that's a grant that Amy got, I think, a year and a half, 2 years ago from USDA. And the goal here is to look at non-traditional irrigation water use on food crops and look at various aspects of that. So this is kind of like, so Conserve stands for Coordinating Non-Traditional Sustainable Water Use in Variable Climates. And again, so it's focused on water reuse and food and health. So the idea here is to address a gap that exists between exploring water reuse in non-traditional irrigation water sources and see how we can apply that to sustainable production for our food supply. The reason why I wanted to highlight that project is because when you want to tackle a problem that is as complex as this one, we always say it takes a village, and it does take a whole town here. And what Amy did here was to basically put a team of experts that were covering as many of the different areas as possible. So that includes bioscientists, engineers, economists, social behavior scientists, law and policy experts, extension specialists, education people, and computer scientists. So the goal here is basically to cover all aspects of water treatment and water reuse, and basically see how we can use microbiome and other types of information to basically change the way we use that water. So this is kind of like just a summary of the approach, so I'm not going to go through the details. So that's taken from the Conserve website, so you can go there and basically take a closer look. But to finish, what I wanted to highlight is that at the end of the day, we can put all our data out there and communicate with a journalist, but what matters really is the public, and if the public is not on board for whatever reason, then basically what we do doesn't translate very well. And this project is definitely a good example of that, because at the end of the day, in this case, and that would apply to other types of built environments, how much people will pay for this food. So if the food is more expensive, then will people pay for it? People might not want to consume that food. So I remember late night shows where Bill Gates was promoting a system to purify water from his own urine. And basically, you know, if people are grossed out by systems like that, then that's something that will never work. So again, at the end of the day, so these are things that we need to consider for all our projects, and social, behavior, and economic factors are definitely something that we need to consider. And so that's basically it for me. So these are the people working in Amy's lab on both that water project and the tobacco project, and people in my lab, and this is the funding. Thank you very much.